Okay. For those who are joining now, we'll be starting in just a few seconds. So hang on for a minute for letting people get logged on. All right, we're going to get started. Hello, everybody. I'm Ken Waterman from FreeThink Technologies, and I want to welcome you to the third of this latest webinar series we have on nitrosamines in drug products. We've already completed two webinars from Maria Ultramari and from Rachel Hemingway previously. Those two webinars are available on our website at freethinktech.com for download, either the slides or the recording. Today's webinar will similarly be available afterwards. And today's webinar I will be introducing in a minute with Steve Baertschi. I want to mention that Gary Scrivens was originally scheduled earlier, but has been rescheduled into November 14th. I want to make a special note that the time is 1500 GMT. It's actually the exact same time in the United States, but it'll be different in Europe because of the change of daylight savings times. So please check Google if you're not sure what time it is for that. But that I think should be a very good webinar on November 14th on understanding and reducing nitrosamine formation in solid drug products. So as part of my introduction, let me just briefly tell you who FreeThink is. FreeThink is a company that we say we think free to achieve more. World-class science to speed better products to market. We include ASAP Prime software and laboratory in our laboratories in the United States for accelerated shelf life determination. We also do a lot of drug delivery development, mainly to solve complex formulation challenges with our recently introduced OsmoCap, an oral osmotic controlled release capsule, as well as solubilization technologies, depots, and other things. Let me tell you a little bit about Steve Baertschi. Steve's somebody I've known for many years. Scary how many years now. And he is one of the smartest people I know, which is I've known a lot of smart people, so he definitely fits that category. He's now the president of Baertschi Consulting, but I believe that's just him, which makes it cleaner and easier. He's specializes in solutions to stability, impurity, analytical, solid state, and formulation issues. Steve was a long-term Eli Lilly scientist, but early retired there in 2015. He got his PhD in organic chemistry from Vanderbilt University. He is known to have organized a lot of scientific conferences, and I've been honored to be a speaker at a number of those, especially on stress testing, stability, photostability, and purities. He's published extensively in the area and is very well known for a book on force degradation that has become the Bible of the field. He's a member of both the American Chemical Society and the AAPS, where he is a fellow. Today's talk will be about the chemistry of N-nitrosamines, insights and implications. As part to make some notes here for people, please type in any questions along the way into the Q&A section. Those will be selected questions will be read out by me, and hopefully Steve will be able to answer your questions. Any questions that we don't get to will be forwarded to Steve, and he'll hopefully get a chance to answer separately. The slides and recordings of this and past presentations will be available in the next few days at the link indicated above, or below, I guess. With that, I'm going to turn things over to Steve and enjoy the webinar myself, and I'm going to go on mute myself. Thank you, Steve.
All right, now I got to figure out how to share the screen because my format has changed and I do not see it, guys. We did this in trial.
You don't see the share?
No, I do not.
Can you go up to the top of your screen or the bottom?
Now I reformatted the view and now I've got it.
Okay.
Okay. No, I want to share screen to. Okay, is it sharing?
You are. You're good.
Okay, good to hear. All right, it's good to be here. I'm going to turn off this video, my video camera in just a second, so you can focus. So I can focus. Actually, I can't even see myself. So you can focus on the slides. I want to thank everyone for. I want to thank Ken and the panel for inviting me to give a talk on this topic, and I also want to thank my Lilly colleagues for. I have heard that there have been a number of Lilly colleagues that signed up for this, and I think the expectations are way too high. People at Lilly are way too smart. Adam McFarland, thanks for setting me up. Now I'm going to shut off my screen and start advancing. We're going to do an overview of the chemistry of nitrosamines, and I'll share the, if I can get the screen to advance, share the outline here. I want to give a bit of a historical context, not a lot, but a little bit. I want to talk about the chemistry. And it's going to be a subset of the chemistry because there's a lot of chemistry that you could cover if you want to do a comprehensive chemistry review. But if you look at some of the references I have, you'll see that you can find a comprehensive chemistry review in the various references that I provide throughout the presentation. We're going to also move into a little bit about assessing the risk. You know, is there a risk? Can a nitrosamine form? And if so, what levels could we predict over the shelf life? And how can we do that? That's just more of a teaser because that's going to be covered more by Gary Scrivens in his presentation on November 14th. I'm going to talk a little bit about the potential for inhibition of N-nitrosation reactions in a drug product. And or scavenging of it using packaging. And I'm also going to talk a little bit about the stability of nitrosamines. So as part of their chemistry, just a little teaser on stability of nitrosamines. And then and then conclude, wrap up, and look forward to a little Q&A. From a historical context, the nitrosatability of amines and amine-containing drugs became very hot topic in the 1970s due to concerns about nitrites in food and the reaction of nitrites and nitrates in food with certain amines. The focus was on the presence of nitrites in foods and the formation of nitrosamines in vivo, mostly in the stomach or in the gut. There was a WHO nitrosation assay procedure developed called the NAP test, and it was designed to evaluate the potential for a compound to get nitrosated in vivo, as well as just in general. But they had both standardized as well as simulated gastric conditions. And there's a few publications you can see down here on that. When they tested a number of drugs using this conditions, where the conditions are taking your drug or your amine at 10 millimolar, exposing it to 40 millimolar nitrite at pHs of 1.5 and or 3 for 1 and 4 hours at 37 degrees, and looking to see if there's a nitrosamine that's formed. And again, you can see how this might be relevant to the stomach, pH 1.5 to 3, 1 to 4 hours. That's kind of a worst case stasis in the stomach, and a 37 degree is the temperature of the body normally. So what it illustrated dramatic variabilities in the potential nitrosatability of drugs, and greater than a 30,000-fold differences in rates for different nitrogen-containing drugs that were on the market. And but it did illustrate the high potential for the N-nitrosation of drugs in the stomach from oral administration. A little bit about the background on what is a N-nitrosamine. I know we've covered this in the previous 2 webinars, but some of you may be first time here listening to me in this webinar. Nitrosamines are N-nitrosamines are the compounds we're concerned about. And you notice I'm not going to talk about the regulatory background. I'm not going to talk about this most recent history very much. I'm not going to talk about a lot of the concerns related to I'm going to focus right in on the carcinogenicity. I'm going to focus right in on what the chemistry of N-nitrosamines. So if you have an amine, a nitrogen attached to another nitrogen, attached to an oxygen, you have an N-nitroso. So the NO is the nitroso. So it's an N-nitroso. And there is an interconversion. These 2 atrope isomers are resolvable by oftentimes, not all the time, but oftentimes chromatographically or by NMR, because this single bond between the 2 nitrogens really has double bond character because of the resonance forms, as you can see here, where there's double bond character and you have a positive charge on the nitrogen and a negative charge on the oxygen. So what that ends up meaning is that you, as I said, you can resolve sometimes 2 atrope isomers due to restricted rotation around that single bond between the nitrogens. When you protonate an N-nitroso compound, it tends to protonate predominantly on the hydrogen, presumably, due to this resonance form, that's where the electrons seem to reside. So adding an H plus is logical to go there. But there is a minor amount of protonation on the nitrogen next to the nitro, attached to the nitroso. And this is implicated in some reactions in acid, typically in strong acid. And you can see down at the bottom here in scheme 2, they talk about protonation of the nitrogen, which then facilitates denitrosification or denitrosation via attack of nucleophiles. And nucleophiles like bromide, thiocyanate, thiourea in the reactions can then denitrosate the N-nitrosamine and lead to a nucleophile attached to a nitroso compound. So the protonation states available affect the chemistry that you see for N-nitrosamines and the denitrosification potential. in our more recent past, after moving on from concerns about in vivo formation, we became concerned about it forming in the synthetic process, and that's due primarily to the discovery in sartan medicines of low levels of NDMA coming from the presence of DMF and a quenching of sodium azide using sodium nitrite, and nitrites and DMF would form NDMA, so the focus was first on the formation of N-nitrosamines in synthetic processes, and this nice paper from Ashworth et al. Describes the biggest risk is in this synthetic process. If you have a high level of nitrite and a trace level of, say, for example, a secondary amine, that's the biggest risk of formation of an N-nitroso compound. Much less risk is when you have a high amine concentration with a trace nitrite concentration. And the least risk is when you have a trace of both nitrite and the amine. And that paper is a good paper to consult for. It's not documented down here. A good paper to consult for synthetic process in aqueous reaction type considerations. More recently, there's been a focus on the formation in the drug product, and this is a concern deriving from the fact that nitrates and nitrites can be found in many excipients at part per million levels. And at first, most of us did not think that was going to be a significant problem, but it turns out that it can be. So the exact source of these trace impurities, I think by now it has been investigated, but from these publications here, it's not completely clear where the nitrites always come from in excipients. LASA has compiled an excellent excipient nitrite database that they published and they continue to add to. I just saw recently that they've updated that nitrite database to have quite a few more results in it, but that is an excellent resource to look and evaluate for your excipient what the typical ranges, levels of nitrite are or may be. Thinking about nitrites and why nitrites are an important consideration, let's think about the nitrosation reaction. So nitrite itself is not a nitrosating compound, but once it's protonated and it has a pKa around 3.37, 3.4, it varies depending on the reference you check, you get nitrous acid. Nitrous acid is a potent N-nitrosating agent, and it typically is invoked, it believed that two nitrous acid molecules combined together, they condense with a release of water to form N2O3, and that N2O3 is the primary N-nitrosating reagent when nitrite is present under somewhat acidic conditions. There are other nitrosating reagents that will be summarized more effectively by Gary Scrivens, I think, in his presentation on November 14th. But you can also imagine, it's also known that NO and can be a strongly nitrosating agent, and the exact mechanisms are not always clear at a molecular level, but once you have nitrous acid, you can get a variety of nitrosating agents that cause this reaction to occur. Now, in the lessons learned document from EMA in June 2020, they say that there have been no reports on the formation of N-nitrosamines in drug products attributed to nitrates and nitrites and excipients. However, this possibility is considered a potential root cause and cannot yet be ruled out. I think we can say that this is not true anymore. There have been numerous reports of the formation of N-nitrosamines in drug products resulting from low levels of nitrites and excipients, not from nitrates, but from nitrites in excipients. So I think we have numerous examples that I'm aware of, and I won't try to name the compounds to go through them, but there are numerous examples where this has been significant. Now, nitrates is one of the things that, if you notice here, they talk about nitrates and nitrites being a concern. Nitrates in drug products, I can say, in my opinion, that are not a concern. They are a concern in vivo, not in a formulated product, and that's because nitrates do not directly nitrosate. They first have to be reduced to nitrite, and that can occur in vivo. That readily occurs in vivo in the stomach. There are enzymes that reduce nitrate to nitrite, but it's not known to occur in drug products. That's a reduction process, and that is it would have to be a very exotic formulation for that to have any relevance. Now, nitrates can depose to nitrites at very high temperatures, example, greater than 100 to 1,000 degrees C. But this does not appear to be relevant to drug product storage and distribution in any sense, and there's no examples of that I'm aware of. It's probably relevant to vulcanization processes, elastomer formation processes. So, various stoppers and elastomers used in packaging may form nitrites or nitrosating reagents from the process of vulcanization, but that's not something that's coming from the formulation itself. Another nitrosating reagent that's relevant or potentially relevant to drug product formulations is chloramines, which typically come from water disinfection processes. So I'm showing the chemistry right here where you have, for example, the tertiary amine that can attack a chloramine. So chloramines are used or are byproducts of disinfection processes, and you can react with the chloramine to form this hydrazine type compound, which then can oxidize and form a nitroso compound. I don't believe, well, and I'll give you some examples here for chloramines from water disinfection processes. In this water research paper from 2011, they looked at these, I don't know how many it is, 25 drugs, and they found 8 of them, I guess it was 20 APIs, and they found that 8 of the APIs were susceptible to N-nitrosation from chloramine during water disinfection. However, the focus of this article is on wastewater treatment before release into the environment. So there's really high levels of chloramines, and there are no examples that I'm aware of from API or drug product manufacturing processes where you might have water that has trace levels of chloramine in it. It's just generally not relevant, but it is something you cannot not rule out. I think in any risk assessment, you need to consider the possibility for the water that you use in either the API or the drug product manufacturing process. But I think in general, it's not a significant risk, and I don't know that there are any examples. There is a possibility, however, of well, I won't talk about that now. That chloride in your API could potentially accentuate N-nitrosation from the nitrite process. An unexpected source of nitrites, nitrite leaching from certain glass HPLC vials. Now, I hope this isn't any, I just grabbed a picture of HPLC vials, and I'm trying to be generic. I don't know of any brand of HPLC vials where this is a problem. I think it's a general glass kind of thing, but there are certain kinds of HPLC vials documented in this publication in JPBA of clofazimine, where storage of the drug substance in when they were analyzing it, so you've got a dilute solution in an HPLC vial waiting to be analyzed, and over time, the nitrite esters of the API were observed after 24 hours, which continued to grow up to 2%. So these are not little trace level peaks. These are pretty obvious peaks. And about 10 to 15% of the vials used in this study would exhibit this particular reaction. And they determined that it was nitrite leaching from the glass of certain HPLC vials. And they also found that you can suppress that leaching and reaction by storing the HPLC vials when it has solution in it at 2 to 8, so in the auto sampler, or if you have a little less acidic diluents. I think the more acidic the diluent, the more extraction of nitrite you get from the or the more reaction they were seeing, the more nitrite esters. So just an interesting thing that it's sort of an artifactual production. It's not really in your product, but you could get tricked into thinking that it is in your product. Another artifactual source of N-nitrosation was discovered at Lilly and expanded upon by researchers at Pfizer, where use of saturated salts for humidity control in when you're exposing samples to, say, for example, an ASAP protocol, or you're trying to control relative humidity. Sodium nitrite is a salt that's used for 60% relative humidity control. And we noticed that there were some artifactual peaks showing up that we couldn't explain in the sodium nitrite that weren't there at any other relative humidity. And exploring that a little bit, we discovered, actually, Lilly researchers discovered that it was from nitrite itself. And you can see here in this example that we published that carvedilol can form in a saturated salt solution where you're not exposing it to the salt solution itself. It's in the, and I'll show you a picture of this in a second. But there's two peaks, as you normally typically see, two resolvable peaks on HPLC artifactually showing up were the N-nitroso compounds. And what it looks like in this cartoon representation, in this humidity chamber diagram, you have a solid sodium nitrite, and then you have water on top of that, and that's a saturated nitrite solution. And there's going to be a certain amount of HNO2 or nitrous acid formation because you've got a certain amount of protons available no matter what the pH is. And that can form low levels of nitrous acid, which can undergo these reactions to form NO gas, which is known to be volatile and then could redissolve on top of your sample. And in this case, we did a trap of water just to see if we were moving nitrosate or NOx into solution, and it was demonstrated using the grease assay that over time in this solution that indeed is what happens. Now, it's probably not just NO that is nitric oxide or NO plus that's being gasified or volatilized, but it's probably it could also be N2O3 or nitrous acid, because those are also fairly volatile. And so it could be a combination of reagents that actually are volatilized and trapped in the trap. The main point of this is to say there are other ways you can see N-nitrosation reactions, and in this case, it's artifactual, and it's a warning for using sodium nitrite as a humidity chamber, humidity controller. And here's the resulting, here's another unexpected source of artifactual N-nitrosation, again performed by Lilly researchers. Dave Myers discovered this and investigated it and found that here's the two peaks that would artifactually form in the analysis of this particular compound API. And he found out that it was an on-column HPLC reaction. So when you use ammonium hydroxide, high pH, and the column has stainless steel frits and acetonitrile is the organic modifier, you would see across a wide range of columns, you would see these artifactual peaks forming, which we discovered to be these two N-nitroso compounds, again, the atropisomers. And we determined that the mechanism of formation is likely that the metal ions from the frit were ablating off the metal ion, depositing on the head of the column, and as the and they were chelating with strong ligands of acetonitrile, which potentiate oxidation reactions of certain metals. And as the compound would pass through the head of the column, it would be retained a little bit and would be exposed to that front of the column where you have these metals that have deposited trace level metals on the head of the column, which apparently oxidize the ammonium or the ammonia to an NOx compound, which then nitrosated the compound. And it's an interesting artifact. But it's documented in the literature now, and it's something that you can be aware of. Since nitroso compounds are such a concern, we wouldn't want to be wondering if it was really in our product when it's just an artifact of the HPLC method. Now let's talk about the chemistry associated with amines. So primary amines are not a concern, but people will say, why are not primary amines? They're going to react with nitrite or an N2O3 the fastest. They're going to form nitroso compounds very fast, but they're also very unstable. So when you react the primary amine with a nitrosating reagent, it rapidly forms an N-nitroso compound, which, because of the presence of this hydrogen here, this proton on the nitrogen, you can tautomerize, which then facilitates the leaving of water and forming of a diazonium salt. And that diazonium salt will react quickly with the water and break down. So you don't have a lingering N-nitroso compound, and that turns out to be make the concern for N-nitroso compounds irrelevant. It makes it not needed. You don't have to worry about primary amines because of that rapid decomposition to diazonium salts. Secondary amines, however, form somewhat stable N-nitroso compounds. Depending on the R groups, it can be very stable or it can be pretty unstable, and it can degrade further, but many secondary amines will form somewhat stable that will hang around nitrosamines and need to be and are stable enough to be to accumulate in the drug product. Tertiary amines and quaternary salts, they sort of form via and nitrosamines can form via a dealkylative mechanism, and even quaternary salts are able to form N-nitrosamines almost at the same level as tertiary amines by a similar reaction. So looking at this, I want to call attention to a couple of things here. When you think of a tertiary amine, there's an initial reaction with nitrite or with the nitrosating reagent to form an unstable nitroso compound. That unstable nitroso compound will degrade to give off HNO or nitrous oxide, and then a nitrous oxide, N2O. And N2O does not cause nitrosation. So you have a decomposition pathway that gives rise to a reagent that does not cause nitrosation, and now you have a dealkylated product. So now you have well, you don't have it dealkylated yet. You have this iminium salt that will degrade and dealkylate by hydrolysis to form a secondary amine and an aldehyde. That's the dealkylation process. And then another molecule of nitrite or of nitrosating reagent can form the tertiary amine N-oxide. And the important thing here is that it really is a 2-to-1 stoichiometry. So it takes two equivalents of nitrite to end up with one equivalent of an N-nitroso compound. And this is important when you're doing a risk assessment for the worst case level that could be formed. It's going to be dictated by that stoichiometry. From a nitrosation rate consideration, acidic conditions are favored for the formation of the nitrosating species from nitrite. So you want it to be in the the low pH region. In order to, you don't want it, but in order for a nitrosating reagent to exist, it needs to be in low pH. But amines are protonated at low pH, so the reaction is actually favored with the amine at basic conditions. And I'm going to illustrate this in the next slide. So this slide's a bit busy, but the next slide will make it visually a little clearer. The N-nitrosation rate is for amines are greatest in the 2.5 to 3.4 region, sort of a competing region between deprotonation of the amine and the formation of the nitrosating species. For secondary amides, the rate is maximized as you continue to go lower. So for secondary amides, the rate is much higher at pH 1 than pH 3. Tertiary amine nitrosation rates are in general much slower than secondary amine, anywhere from 100 to 10,000 times slower than their analogous secondary amines. And apparently N-nitrosation only occurs in tertiary amines if there are protons alpha to the nitrogen. I don't know for sure if this has been completely tested, but I found this in one of the references I have below. Here's the visual representation of that Gary Scrivens has put together, and you'll see this again in his presentation on November 14th. The fastest rate of N-nitrosation is in this pH range between in the 2.5 to 3.5 region. And where the y-axis is percent unionized form of the nitrite, and the unionized form of the amine is in the orange. So the pKa's of the amines are typically up here. And one of the take-home messages is, so it's a competition between deprotonation of the amine and protonation of nitrite. And one of the important things is that if you have two amines and one has a higher pK than the other, it's going to be a slower nitrosation reaction in general. So the higher the pKa of the amine, the slower the N-nitrosation reaction in general. At high pH, you can achieve, so at high pH, generally you see very low rates of N-nitrosation because you have so little nitrosating species. It's going to be nitrite, which does not nitrosate. But if you have high levels of aldehydes around, say for example formaldehyde or chloral at very high levels, it can increase the rates. So in this graph right here, the yield percent in a reaction study goes way up in terms of N-nitrosation formation. Even at high pH, it's much higher with formaldehyde than without formaldehyde. It's a very low reaction above pH 7 without an aldehyde around, but with an aldehyde around, it's substantial. And it proceeds through this mechanism where reaction with an aldehyde forms this iminium compound, which nitrate can react directly to form a nitrate ester, which then rearranges to form an N-nitroso compound. It has been concluded by most people that this is not probably relevant to solid state formulations because you need such a high level of it. But Paul Harmon has proposed that this may be relevant to solid state formulations because of a localized formation of formaldehydes in degradation of certain excipients that can be fairly high. So that's something that remains to be tested, but it's important to. It may be good to know that it could be relevant to a solid state formulation, which can be tested. And I hope people will be testing this or have tested this and will publish on it soon. Assessing the risk, ranitidine is an example where there was no necessarily an N-nitrosating agent. It was just ranitidine itself. And you see this tertiary amine, which apparently degraded to form dimethylamine and then got N-nitrosated to NDMA. And if we look at the mechanism was not straightforwardly figured out. And they did an elegant study, some GSK researchers did an elegant study to determine using labeling that indeed the tertiary amine was what was N-nitrosated to form NDMA, and that the the compound, the nitroso functionality came from the nitro group that was attached to the molecule. And how that actually happened was not clear. More recently, Harman has proposed that it could actually be from two oxidation steps. So let me show you what that means. Two oxidation steps where ranitidine he proposes that peroxy radical oxidation of this benzylic position, which should be a highly activated hydrogen, that could be oxidized, abstracted, oxidized to form DMA. And at the same time, a peroxy radical could also oxidize this functionality where the nitro is attached. And if you oxidize this and release the aldehyde, you can release a nitrite ion. And at that point, you have nitrite and DMA that's formed that could react together to form the NDMA. The implications of that would be that Paul Harman suggests is that there may be a strategy for inhibiting that reaction using desiccants and oxygen scavengers, which I think remains to be tested, but is an interesting hypothesis. And I would think that people have already been working on testing this to see if it's actually true and can be used as a control strategy. Now, you also notice that the functionality here has a furan with a benzylic position attached to a nitrogen. When you have that kind of situation where you have a benzylic active next to a nitrogen, a tertiary nitrogen, it can be set up to disconnect, to dealkylate and form NDMA rather readily, and that's the case with ranitidine. So you can imagine an N-nitrosation reaction could also trigger the release of NDMA, so a direct reaction and then cleavage. Irbesartan is an interesting one. I'm about 5 minutes from finishing, so those who are tracking, I will skip through a few slides at the end, but I just let you know I'm paying attention to the time. Irbesartan was in October 2021 was recalled in the U.S. market because of the presence of N-nitroso-irbesartan. And it turns out that what was believed to be N-nitroso-irbesartan was not. It was actually the oxime derivatives, and they were isomeric. And so the reaction was presumed that it was an N-nitroso, and actually these oximes are not near as toxic as the N-nitroso compounds. The reaction mechanism that the researchers published appears to be forming of an enamine. So you've got kind of a nucleophilic site in this position right here that can react with the nitrosating reagent to form reacting with nitrous acid, which then can rearrange to form this these two oxime derivatives, and not even involving the nitrogen that could form a N-nitroso compound. So it's important, just because you've got the right molecular weight and you've reacted it with something with nitrite, doesn't mean it necessarily is an N-nitrosamine. It's good to understand the chemistry and using irbesartan as an example of when it was misunderstood, leading to serious ramifications of product recall. In October 2020, rifampicin and rifapentine the FDA became aware of these N-nitroso impurities that were formed or found in those products. And I'm not sure of the mechanism, how they concluded it forms, but just from a simple degradation pathway, you can see that this hydrazine imine could readily hydrolyze to form a hydrazine derivative, which could oxidize upon storage to form an N-nitroso compound. So I think whenever you have something that can hydrolyze to a free hydrazine, you should be concerned about the possibility of forming of an N-nitroso compound from simple oxidation and no presence of an N-nitrosating reagent. There are a couple of approaches to determine whether or not your compound, an API, can form an N-nitroso compound. The original NAP test has some problems with drug solubility and artifact temp formation. but there have been two approaches that have been developed. One is called the modified NAP test that was published by some researchers, where they modified nitrite to nitrite, and they did some offer some solvent combinations. And in just 50 minutes at 37 degrees C, you can see if your compound is likely to form an N-nitroso compound. And the conclusion that they propose, if no N-nitrosamine is formed under this condition, there is no realistic risk to formation in your drug product. Similarly, I think people from the, and I'm going to need to stop pretty soon. Let me finish up this one slide. These three approaches from Ashworth et al. At exposing a compound to
Steve, are you there? Somehow we lost Steve. Hold on, everybody. I don't know what happened, but somehow we lost Steve. Hopefully he can rejoin us in a second. I guess I get to fill in and try to answer questions. While we have a minute, please put in questions into the Q&A, and when we get Steve back, we'll be asking him those questions. Also, remind people that the slides and the recording of this presentation will be available. The recording has the ability to have closed captions for those who need that. So please go ahead and put in questions, and we'll try to get them answered as best we can from this. So a couple of questions that were asked, I'm going to try to bring up while we're waiting for Steve to hopefully get back. One was why this issue has come up recently, and I probably will try to take a crack at that. I think there was some work involved in detecting the dimethyl and nitrosamine from ranitidine, I believe was the first of the ones that became a concern. And I think that some of this is once you detect it, you can't ignore it. And so I think part of this was once it was detected, the regulatory agency started to say, wait a minute, is it in more things? And as people found these extremely low levels in more things, it became more of a concern. I'm looking to see some of the other questions. Somebody saying, please bring back Steve, and I agree. And so I want to ask Steve a lot of these questions. We're trying to get him back. So hopefully we can get him back in a minute. Maybe I'll try asking my panel here if any of them could answer a question. Perhaps Rachel, putting you on the spot, what is the view on aryl nitrogens as precursors for nitrosamine impurities?
Yeah, I mean, there's some evidence that was it aryl and indol Yeah, as sort of activating groups, that there is some evidence that they can sort of be precursors to nitrosate. I know we have a little bit of coverage on that in Zenith. Certainly not as higher potential, the kinetics is lower, but yeah, I think there's still
So are you,
a
just
a
so I know,
requirement to the concept to sort of do an assessment of those kind of compounds.
are we talking aryl, the nitrogen in an aromatic, or nitrogen or an aryl substitute on an amine?
Yeah, I think I was thinking more of a substitute on an amine. I think that can be more of an activating group.
Things like pyridines or other nitrogen ring compounds, I'm assuming, are less likely, correct?
Yeah, I definitely think so. I think the actual nitrogen in the indoor ring, for example, I haven't seen any
That's what I
literature, any evidence that those directly nitrosate, but I think they can be an activating group. Yeah, on
if they're one of the substitutes in like a secondary
exactly.
amine, an aryl substitute would certainly seem like a good candidate for that.
Yeah,
Thank you.
I was just having another look through. There was one on excipients that I thought I could maybe
Mhm.
where was it?
We're getting
Oh,
Steve back.
back.
Okay, Steve, are you back? Steve,
Yeah, sorry, my computer went through a whole reboot on for some reason.
all right. I think you need to wrap up and we'll start asking you questions. Okay?
Why don't we just consider it a wrap?
Okay.
And I'll
Let
ask
me
questions.
start throwing questions at you, Steve. Thank you again, and wonderful talk. And the full talk will be available for anybody. The slides and everything. Um, so first question: has anyone used primary amines as a scavenger? In other words, how does the rates of primary amines compare to secondary? Could you use this as a scavenger against the nitrite or nitrous oxide, nitrous acid, sorry?
Yes, you could, and it has been done. I think in the research by Nanda et al., Nanda Kaushik et al., J Farm Sci in 2021, they showed the potential use of glycine, lysine, and histidine. And I think Gary Scribbins is going to talk about the use of some research with cysteine that worked very well. So I think that amino acids are very on the table in terms of potential very good inhibitors.
Thank you. One of your slides, you mentioned secondary amides rather than secondary amines as reactive. Are secondary amides reactive, or was that just a typo?
They're not reactive. They're not practically reactive in a drug product formulation. They are synthetically, you can make N-nitroso compounds under extreme, very acidic conditions and under the right conditions. So there's a number of other amine containing compounds that you can make N-nitroso compounds from, but they're not very relevant to drug products and APIs.
Okay, so that was probably a typo. Their amides are not really risk.
Was not a typo on my part. It was amides nitrosate at very acidic conditions. Their optimum is not between 2.5 to 3.5. Their optimum is down at 1 or below.
Okay, next. Can you clarify that you said dealkylation and subsequent nitrosation of tertiary amines occur only in the presence of alpha hydrogens? Is that correct? Clarify. And
that's my understanding. I got that from a literature article. I think I documented it in the slide, but I am skeptical. I'm not completely sure about that, the truth of that.
Okay. Is there any evidence that nitrite is oxidized to nitrate in stability studies?
That's a good question. Can nitrite be oxidized to nitrate? Um, I I would think that that would be very possible, and that could be the nitrites over time seem to decrease in excipients, the levels over time and storage. And what they go to, is it volatilization? Is it oxidizing to nitrate? It's not clearly defined as I understand it, but nitrites are not completely stable in formulations, and they will decrease over time. So when you test a fresh batch of excipients or product, you're testing the worst case scenario for potential nitrite levels.
Okay. Do you have kind of an order of magnitude of risk, how much nitrosating agent would need to be present to account for a worry of secondary amines in a drug product?
There's a publication by Galloway, for Galley, I can't remember, where a publication that discusses the native form of nitrite that's typically present in excipients, where they assert that it's ammonium nitrite, and they claim that their projections are only 1.6% roughly is a worst case amount of reaction of the nitrites naturally present that will react to form N-nitroso compounds. I've talked to Gary Scrivens, and he's going to talk about this a little more, I believe, in his presentation. I think it's some small percentage, maybe 10% of the nitrite present could give rise to N-nitroso. They tend to plateau. The question is, what are the limits that you're worried about, and how much nitrite? So I would say between 1 and 10% of the nitrite present is could be considered to give rise to that level on a molar form of the N-nitroso compound.
It's an interesting arithmetic. I'll have to think about that too.
Yeah, so it's a molar basis.
The trouble is you're sensitive. Sometimes we're worried about parts per billion or parts per million, so it doesn't take much, unfortunately.
Right, and I don't have a ballpark level in my head, but 1 ppm is a typical amount of nitrite that's in a typical formulation, and that's enough to cause a problem.
Because of molar equivalence against the drug, that doesn't take much. Yes.
That's enough to cause a problem. However, now with the new CPCA levels, the read across, the levels have moved from 18 nanograms for many drugs up to 400 or 1500 nanograms formation. And in those cases, it may be irrelevant. In other words, the worst case scenario may not cause a problem, but it's a mathematical question.
Okay, and I'll try one more question. The impact of relative humidity, one of my favorite things. What's your anticipation as activation of nitrite requires dehydration? Does water activity stabilize or destabilize?
I can tell you that Gary Scrivens is going to talk about that very thing. And it is an important factor, the relative humidity, and it's a factor in how fast nitrite will degrade too upon aging. So you could imagine aging your excipients for a little bit and significantly decreasing the amount of nitrite available to nitrosate. But I don't know if anybody's taking that kind of approach.
And just want to mention, somebody brought up and see what your thought is on atmospheric nitrites or NO compounds, let's say, especially in environments that have a fair amount of NO compounds in the air, air pollution, let's say. Is it your anticipation that that contributes or not to the issue?
That again is a good question. I think NOx would have to be a pretty bad scenario, and I have read of some scenarios when it's super humid and the NOx is very high in an urban area where manufacturing processes have trapped nitrates and nitrites. That was before consideration of nitrosamine concerns. But I think that would be probably in a manufacturing environment. I haven't seen any relevance to actual real world data, but it's a good question that I don't know.
I've heard it reported, but I haven't seen the publications either, but I have heard people say they believe that that's consistent with their data, but
Yeah.
okay. I think we're out of time. I want to thank Steve again for a wonderful talk and remind people that this presentation, the slides, and all of this are available for those who want. Let me again show you where well, I don't have it up, but it's on the FreeThink website. There's resources and webinars. You can find that and get the information, the recordings and from this and the previous slides. Thank you everybody for your attention. Look forward to the next webinar in a couple weeks. Bye everybody.